How's everybody doing this morning? It's Friday. Woo! So I don't need to remind you, but I will remind you. We have an exam on Monday, and the format of the exam is the same as the format of the exams I've done before. I wrote the exam last night, and I'll even tell you a little bit about what's on it. So the first section is as before. It is a short answer section. Usually can be answered in one or a few words. It doesn't take very much. There are 13 questions in that section. Each one's worth 3 points, except for one of them is worth 4 points. That's a total of 40 points in the first section. Okay. The second section is a problem-solving section, and there are 2 problems, and each one is worth 15 points. That's worth a total of 30. The third section, I actually shortened because this is the exam where students say, oh, I don't have enough time to do stuff. So I shortened the exam and made it 2 questions, not 3, not 4, but 2 questions, and each question also worth 15 points. Okay? One of the things I'll tell you is the first exam is usually the one where students say, oh, I don't have enough time to do stuff, etc. And so I shorten the exam, but I also tell you that when I shorten the exam, that means that each question becomes worth more points. And so it's a balance. Students say, oh, I don't want to have a question worth a lot of points, etc. But we can't have it both ways, unfortunately. So I decided this time to shorten it and make that, less of an issue of time. Nonetheless, I still think that you need to be careful with your time. So don't spend too much time on one problem, and budget your time accordingly. All right? 40, 30, 30. Okay? So the point balance is 40, 30, 30. Let's see, what else I'm going to say. So don't spend too much time on it. Don't be afraid to raise your hand and ask me a question about. I can't, of course, give you the answer. But if you don't understand something or don't understand what I'm asking about something, raise your hand. You'll see that I'm the person that answers all the questions, so I get a jogging exercise literally running through this room. And if you want your questions answered, don't wait till the last minute, because if I have 20 of them at the last minute, you can't do that. Okay? Now, you'll see I'm very, very protective with time, and that's basically to protect everybody. So what will happen is at the end of the class period, I'll say, okay, everybody needs to stop, and I'll give you warning beforehand, and at that point, you cannot put a pencil back on your exam. If you haven't written your name on the exam by then, okay, I'm sorry, but I've warned you. Okay? And the reason for doing that is what I discovered in years past is some people, to try to get that little extra edge, would get way to the end of the line to turn their exam in, and they'd be writing as they're going up the thing. And that's just not acceptable. So when I say stop, everybody stops, which means everybody gets 50 minutes. Okay? And hopefully, and again, I don't think time should be too much of a problem, but hopefully it won't be a problem for you. But when I say stop, you need to stop. Otherwise, I will penalize you. Okay? And that's, again, to be fair to everybody, not be mean. I don't like being mean for any reason. Okay? The other thing is, the sooner you get in and get organized in the room, all right, the sooner we can get the exams out and get things going. So we won't have a situation where we have to go less than 50 minutes. I'll have the time showing on the screen, so you'll be able to see how much time there is, etc. And what I would ask you to do when you come in is as follows. You need to sit in every other seat, which means that not everybody's going to fit down here. Some of you are going to need to sit up there. But the way to organize it is as follows. This row right here will become seat number 1. Okay? So you sit in seat number 1, you sit in seat number 3, you sit in seat number 5. Okay? And if there's a go all the way over here, there might even be a seat number 7. All right? Just because somebody's sitting in seat number 2 doesn't mean I want you to sit in seat number 4. If they're sitting in seat number 2, I'll say, get up, move, okay? You can't count, all right? And because I can't have you sitting adjacent to each other, all right? Makes sense? You say, well, I need a left-handed seat. Well, if you need a left-handed seat, find one, okay? Or that's a left-handed seat, and I'm right-handed. We'll find another one, okay? But the important thing is I need you in 1, 3, 5, 7. We'll start counting this one from this side. And if you're looking for left-handed seats, this row has a decent number of left-handed seats on it. So we go 1, 3, 5, 7, 9, 11. You guys can do the math. Okay? And don't think because 11 is here, well, I can sit on 12 because it's in the end. I don't want you to do that. I want you 1, 3, 5, 7, 9, 11. Okay? And again, that saves us time. The more of you I have to say, please get up, please move. What was it? This seat's broken. Well, then, I can't have you sit there. Then we'll start this one right here with number 1 again. So 1, 3, 5, 7. Okay? And it does take time to come in and get settled. And it's a problem sometimes if the previous class is having an exam, because they run over and they affect us, etc. So the sooner you can come in and get seated, the better off you'll be. Okay? The same is true up there. So we'll start 1, 3, 5 going that way, 1, 3, 5, and then 1, 3, 5 going over this way. Make sense? Clear as mud? Okay, so second announcement is I have scheduled a review session, and the review session will be held tomorrow on Saturday, starting at 4 p.m. in ALS 4001. ALS 4001. I will videotape that, and yes, I will try to repeat the questions as I get them so that people who aren't there can hear what the question was as I'm doing the answer, etc. And there's no format. It comes, I answer your questions is what I do at that. Okay? Now, I expect there will be questions. Okay? Please don't come and say, well, I don't understand such and such. Put it in the form of a question. I really want to answer questions for you because I can't go back and give 15 hours worth of lecture over things as I've already done. All right? But come with questions. I'm happy to answer questions for you at that. Okay? Make sense? Everybody understand what's going on? Okay, so let's see. I think that's all I had to say in terms of announcements. So we now turn our attention back to biochemistry. You remember the material for the exam is what I covered through Monday. So at the end of the highlights on Monday, that's where we stop, and you'll see that marked on the online thing. Okay. We turn our attention now, we've learned something about how enzymes work in general, and we've learned very basic principles like allosterism. Okay, and now we're going to turn our attention to looking at some specific enzymes and specific things that they do in their catalysis. So we're going to sort of get a little more up close and personal with the enzymes as we go through and understand them. We're going to talk today something about mechanism. And mechanism, I'll be honest, is not my favorite topic. It's probably not going to be your favorite topic. But we need to understand something about how mechanism works for one or two enzymes. And I think you'll see some common things starting to emerge once we've done that. So I'll ask you to just kind of bear with me as I'm going through some of the mechanism work. Okay, well, the first group of enzymes that I want to talk about are things that we've already said a little bit about already, and these are the proteases. Proteases are enzymes that break peptide bonds. They break peptide bonds. Okay? A protease will break down proteins. Will a protease break down other proteases? Yes, it will. And the reason we have proteases is because it's important in our digestive system, among other places, but it's important in our digestive system for us to be able to degrade the proteins that we have consumed so we can get the amino acids out of them and then use those amino acids to make our own proteins. Okay? Here is a schematic showing the breaking of a peptide bond. There's a peptide bond right there. You'll notice that what is required to break a peptide bond is water. This is a hydrolysis reaction, so all proteases are adding water across the peptide bond. And the net result of that is we started with one long peptide and we end up with two shorter peptides, broken as you see right here across here. Now what we're going to see as I go through this is how that peptide bond gets broken for one class of proteases. And I won't worry about that, you've seen that before. All right. Chymotrypsin is a protease, and it's the protease that we're going to focus most of our attention on. It's a protease that cleaves adjacent to several amino acid residues. The amino acid residues it cleaves adjacent to tend to be fairly hydrophobic. Okay? That's the common tendency. All right, so we can see here's phenylalanine. Chymotrypsin would cleave right there where that red bond is. It would also cleave adjacent to a methionine, and you can see right there where that red bond is. Now the problem of studying the cleaving of peptide bonds is it's hard to see that happen. Okay? It's hard to see it happen. We don't, as I like to say, biochemists are lazy people. And they like to have simple things that they can study. And it's possible to study this, but it's a very tedious process to watch the breaking of that peptide bond. And so we like to find easier and simpler ways of studying basically the same reactions. Okay? I forgot I was going to say this too. And what I was going to say is that chymotrypsin is a serine protease, meaning that it uses serine in its catalytic activity. And so last time when I talked about the chemical DIPF, which can bind to serine residues, if a serine residue is in the active site and it's required for catalysis, then treatment of DIPF of an enzyme will inhibit that enzyme's ability to function. So if I take chymotrypsin and I treat it with DIPF, the DIPF will, in fact, combine with serine and make that enzyme completely inactive. So that tells us that there's a serine that's active in the active site that's necessary for the catalysis. And chymotrypsin is, again, what we refer to as a serine protease, meaning it uses serine in that catalytic activity. Well, here's the lazy thing I was going to talk about. The lazy thing I was going to talk about is that, as I said, it's not easy to follow peptide bonds. But it turns out chymotrypsin will cleave a lot of things that look like peptide bonds, and one of them is this molecule up here with this mouthful of a name that you're not going to memorize. Okay? Or the structure or any of that. The only important thing about this compound is that this compound, before it's been acted upon by chymotrypsin, is colorless. But after chymotrypsin has treated it, it discovered that one of the products is yellow. Well, color is something that's very, very easy to follow in the laboratory. You simply put it into a spectrophotometer and watch the absorbance change over time, looking for absorbance of yellow, and voila, you now have a compound that is that you can study. So instead of studying peptide bonds directly, what we study in many proteolytic reactions are things like this. How many times do I have to do this? I think I must be the only person that ever changes this battery. I should just do it before I start. Okay. It took me many years to learn that changing the battery actually made that sort of weird sound be less bad. Anyway, all right. So it's not my day. Okay. So anyway, what we're interested in studying with chymotrypsin is basically the production of yellow color when chymotrypsin is mixed with this chemical. The more yellow color, the more enzyme activity we see. All right? Very simple thing. All right. Well, we're not really so much interested in yellow color as we are interested in understanding what chymotrypsin does and how it does it. Now, when people started studying chymotrypsin, they discovered something very surprising that they didn't expect to see. And what they saw was this. They saw that chymotrypsin, when they mixed it with this chemical, at the very beginning, looking very quickly, you notice milliseconds after mixing, all right? That's thousandths of a second. Very quickly, very early on, the rate rose very rapidly, and then it leveled off. And you might say, oh, well, that's the V versus S plot. But we're not plotting V versus S. Notice we're plotting time. We're plotting velocity versus time. So the velocity is actually changing over time. Now, that was very difficult to reconcile. And the velocity changing over time, when this curve first was produced, smart people looked at that and said there's really two steps in this catalysis. A step that occurs fast and a step that occurs slow. So what we're seeing is this fast step occurring and then the slow step occurring. Okay? Well, what does that mean? All right? Well, what it means is this. If we look at chymotrypsin's action, what we discover is that chymotrypsin, it does have two steps to its catalytic activity. All right? The first thing that happens is the enzyme, okay, it breaks the bond that releases the yellow color. So here we are, we've got this original molecule. Here's the bond broken, and when the bond is broken, that's when the yellow color is produced. Okay? The net result of that is that not that this other piece that's produced by the cleavage hangs around and does nothing. It turns out that that other piece is covalently linked to the enzyme for a brief period of time. Okay? So the fast phase is breaking the bond to release the yellow color. That leaves you with the other piece covalently bound to the serine in that enzyme. And then the slow phase happens with the removal of that other piece from the enzyme. Okay? And you're saying, oh, that's suicide inhibition. No, it's not suicide inhibition because it's not inhibition. The other piece gets released from the enzyme. That covalent bond is only a transient bond. But the time it takes to remove that piece from the enzyme is longer than it takes to cleave that first molecule off. Everybody got that? 2 steps. A fast step that cleaves the bond, releases the yellow, and then a slow step that releases that other piece from the enzyme. Well, as I said, this other piece becomes covalently attached to the serine residue. Okay? Let's look at this in terms of what the enzyme is doing. Well, actually, let me back up on that. You know how I like structure, okay? We're going to look a little bit at structure, sort of schematic structure today. But this shows the serine in the active site, and the serine in the active site is at position 195. You don't need to know that. But I show you this to show you that it's internal to the enzyme. And that region internal to the enzyme is going to be important for understanding the catalytic activity of chymotrypsin. All right? To understand the catalytic activity of chymotrypsin, we have to understand the roles of 3 different amino acids that are located in the active site. 3 different amino acids. The 3 different amino acids are referred to as the catalytic triad, because all 3 play a role in catalysis. The numbers beside them refer to their position in the polypeptide chain. That is their primary sequence. So histidine is number 57 in the polypeptide chain. Aspartic acid is 102, and serine is 195. Notice that they are all close to each other in location, but they're not within 10 amino acids of each other in primary sequence. What does that tell us about structure? This is tertiary structure, right? Okay, so we're seeing tertiary structure. We've seen folding, and the folding has brought these things into close proximity with each other. All right. Now, each of these, as I said, has a role in the catalytic process. All right? Serine plays the most visible role, but it turns out that the other 2 have very, very important roles as well, and I'm going to explain that. The most visible role that serine plays is it becomes ionized. Several of you have asked me, how come we don't see serine ionized? It's got an OH group, there's no pKa for it. And I told each of you it's because it takes a very high pH in order to pull those protons off, and it's way beyond being practical for us to study in a laboratory. Okay? So we can't ionize serine, we can't ionize threonine very easily under normal conditions. But here you see serine is actually losing a proton and becoming ionized. And it's becoming ionized because of action of specifically this histidine, which is right next to it. Okay? This is going to be important because it turns out that this alkoxide ion that you see right here is very, very, very reactive. The formation of this alkoxide ion is a critical step in the catalytic action of chymotrypsin. This alkoxide ion actually attacks the peptide bond. Okay? So we have to understand, therefore, how the alkoxide ion forms in order to understand how chymotrypsin does what it does. So that's what I'm going to talk about next. But everybody understand, the alkoxide ion is very, very important, and it's produced by the removal of that proton. Notice how that proton is pulled over here by the histidine. Well, how does this happen? Okay? How does this happen? If it takes a lot of pull to get that thing off of there, how does it come about? That's what's shown in this catalytic mechanism. Now what I'm going to do with this catalytic mechanism is I'm going to sort of explain it to you in sort of generic terms. I'm going to go all the way through it, and then I'm going to come back and go through it again. So if you don't get it the first time, don't sweat. I will go through it again. And the common question I get with this mechanism is, well, do we have to memorize the mechanism? And the answer is no, you don't have to memorize the mechanism. But there are certain very important steps in the mechanism that I think you should have some understanding of. And I'll point those out to you as we're going through it. This won't be a mystery, I don't think. Okay? Okay. Well, here's chymotrypsin. We can see the catalytic triad. We're looking at the active site. We're going to see, in looking at this enzyme, I'd like to describe the region of the active site in sort of visual terms. Remember that the active site is in the internal portion of this protein. I like to think of the place where this is located as part of a cave. Part of a cave. Anybody here a caver? Anybody climbs around in caves? I've got a friend that's a cave. You are? Okay. So you've been in caves. You've got these little pockets. You've got these little rooms off of here and so on and so forth. All right? And these little rooms that are off of this cave are going to be important for us to understand. Okay? So the catalytic pocket is basically a room of a cave, and it's going to have other little rooms sticking off of it. Two specifically. Okay? All right. So we're in the cave right here. We're looking at the active site. There's the aspartic acid, there's the histidine, and there is the serine. You can see that what's happened up here in red is that the substrate has bound. Now the substrate, you remember, is a polypeptide. And you remember that the polypeptide gets cleaved at a specific place. The enzyme doesn't cleave adjacent to every amino acid. It only cleaves adjacent to certain amino acids that I said tend to be fairly nonpolar. You saw phenylalanine was one of them. You saw methionine was another one. Okay? Let's say this enzyme binds to a protein and it gets adjacent to the active site, it gets the wrong amino acid. When that happens, nothing happens. It lets go of it, it goes away, and everything is fine and dandy. When the enzyme binds to the polypeptide chain and it gets the right amino acid in there, something magical happens. The magical thing that happens is now everything is positioned properly. The enzyme undergoes a slight conformational change, just like you've seen with hemoglobin and just like you've seen with other proteins that I've described. A very slight conformational change. That very slight conformational change is what causes the alkoxide ion to become ionized. And I'll explain how that happens in a second. So unless you bind the proper substrate in there, that is the proper amino acid to be cut next to, the enzyme doesn't do anything. It just lets go of it and comes back and goes away. When the proper one gets there, then there's a proper rearrangement of the polypeptide chain, and now things start to happen. Okay, so let's think about what things are starting to happen. That slight conformational change causes the geometry between aspartic acid, histidine, and serine to change very slightly. The first thing that happens is you notice aspartic acid is negatively charged. Okay? The negative charge on this aspartic acid carboxyl group, being moved closer to that ring of histidine, causes those electrons in the ring of histidine to move away from where that negative charge is. So we can think of it in these terms as the electrons, instead of being evenly distributed across this ring, move to the right side of the ring. Notice that to start with, this nitrogen does not have a hydrogen on it. That makes this nitrogen relatively electron rich. This nitrogen is relatively electron rich. Is that a hydrogen bond? You betcha. If I add some negative charge to this nitrogen, is there going to be a stronger attraction for that hydrogen that's sitting out there? You betcha. And so what happens is this nitrogen abstracts or pulls off that hydrogen off of that serine hydroxyl group. Now the figure doesn't show it here, but that creates the alkoxide ion that you saw in the last figure. So binding of the proper substrate moved aspartic acid close to histidine. Histidine became relatively negatively charged. That negative charge was concentrated inside of this nitrogen, and that pulled off that hydrogen from the alkoxide group. You'll notice here it says positive, and it's now positive because it's already pulled this hydrogen off. Hydrogen is positively charged. In the process of that, this guy was partially negatively charged. It's the hydrogen that made it positively charged. So this mechanism, they really need to have one more step in there showing the removal of that hydrogen, which they haven't done. Everybody understand that? Questions? That's the most critical step, actually. So the proper substrate binds, we get this reaction occurring. If the proper substrate doesn't, if there's some other substrate that binds, nothing happens. And that explains why we don't see chymotrypsin cleaving all peptide bonds. This change only happens when the proper substrate binds to it. All right. Well, here at this point we've done, what happens is that this alkoxide ion is very, very reactive, as I said. And what it does is it attacks the peptide bond right next to where that amino acid had bound. It attacks it. And when it attacks it, it makes a peptide bond of its own. It makes a bond of its own through here, not a peptide bond, but a bond. So we see this attack of the negatively charged oxygen right here at the peptide bond. And the product of that reaction, and I'll explain Oxyanion Hole in a second. The product of that reaction is a broken polypeptide chain. Here's one piece. Here's the other piece. The one that's covalently attached is on the right. The one that is free is on the left. This guy on the left goes floating away. That's the very rapid phase of the catalysis. In the meantime, we now have that O on the serine covalently linked to the other piece. This is now going to be the slow step in the catalysis. We're not done because we have to get rid of this other piece that's attached to the O. Yes, ma'am?
Was it R1 or R2 that was the non-polar side?
Was it R1 or R2? So R1 is cleaving on the carboxyl side of R1. Okay? Okay. We've released R2. We now have the other portion of the polypeptide chain that's covalently linked to the enzyme. We have to get that off before this cycle is done. And this is the slow step. Okay? I'm going to come back and talk about Oxyanion Hole. Don't sweat that for the moment. All right? It's not as big of a deal as it would seem. This is one of the other rooms off of the cave. All right, well now we have to get rid of this guy. And to get rid of this guy, it requires water. Remember that the breaking of a peptide bond requires water. Now we're seeing water coming in, and water is needed to release this other polypeptide from the enzyme. How does that work? Well, water comes into the active site. We see water positions itself. Look how it positions itself. Here's a hydrogen, here's an oxygen, here's a hydrogen. This nitrogen, which is now once again partially negatively charged because the proton has gone away here. The proton that it took has now gone away here. This guy is partially negatively charged again. What do you suppose it's going to do with that hydrogen? It's going to pull it. And it's going to leave a reactive hydroxyl group. That reactive hydroxyl group is going to attack the carbon that's attached here. It's going to form a structure in this Oxyanion hole that I'll talk about in a minute and cause it to be released. Now, the only things that have to happen are the enzyme has to go back to its original state. It releases this polypeptide here, and we're back where we started. You'll notice that in both steps, a reactive oxygen intermediate was created. In the first case, the reactive oxygen intermediate was created on a serine residue that attacked the peptide bond. In the second case, a reactive oxygen was created on a water molecule, and that attacked the other portion of the polypeptide that was linked here. Okay, now I've gone through it once. I haven't told you about the Oxyanion hole, but I want to say or ask for questions if you have questions, and then I'll tell you about the Oxyanion hole. Yes. I'm sorry. Say it again. Yes? Yes. Yeah. So she says the book talks about nucleophiles. Can I define that? Absolutely. A nucleophile is an atom that is electron rich. It's attacking a nucleus because it's got these extra electrons. The alkoxide ion is a nucleophile. So a nucleophile, okay, is electron rich. Notice it's a negative charge. O- is a very reactive molecule. That very reactive molecule is what's causing this bond to be broken. This reactive oxygen down here, this hydroxyl, is also a nucleophile. It's very electron rich. It's OH-. Okay? It's attacking this guy here. And we're set. Make sense? Yes, sir.
On step 4 there, how does the part of the polypeptide chain leave from the nitrogen?
There's nothing holding it.
Nothing holding it.
Because this is basically a hydrogen bond. Okay? In the process of breaking this guy here, this guy falls onto that hydrogen, and the nitrogen lets go of it.
Okay.
Okay? So there's nothing holding it in place at that point. It looks like, well, it's got to let go of this too, but that's only a hydrogen bond. That's not a covalent bond. Okay? Question up above. One and two. Okay. I'll go through that again in just a minute. Okay. Another hand I saw. I'm sorry. Say it again, Ariel. So the fast step is the breaking the bond. This step right here, rapid. This step, releasing this guy, very slow. Okay? So his question was, which is the fast step, which is the slow step? Okay. Well, let me say a word about the Oxyanion Hole, and I'll give you 2 rooms that are off of this cave. Okay? They're just in the same region. They're just a little pocket of the enzyme. So we can think of this cave, and there's a little pocket and a little pocket that's off of this cave. That's all that we're talking about. Okay? One of these pockets is called the Oxyanion Hole. And all it is a little portion of the protein that stabilizes an unstable structure. It stabilizes, I'll explain why that's necessary in a second, but it's stabilizing an unstable structure. Okay. That unstable structure arises as a result of the attack of the oxygen on that polypeptide bond. On the peptide bond, okay? So, as the oxygen attacks this carbon, and by the way, there's the nucleophile, there's the nucleus it's attacking the nucleus of that carbon. As it makes that attack, this intermediate, you see the electrons up here, the electrons are shifting all around. We see a lot of movement of electrons. This guy right here is extraordinarily reactive. If the enzyme does not protect this reactive intermediate, what will happen is it'll react with the enzyme, and then you've created a suicide inhibitor. It's not going to let go of it. So the Oxyanion Hole is there to stabilize an unstable intermediate so that it doesn't react with the enzyme and become covalently attached to it. As a result of that stabilization, what happens is instead of reacting with the enzyme, the molecule falls apart. That's what breaks the peptide bond. That's how we get from here to here. The enzyme, this intermediate, this very reactive intermediate is falling apart, and as it falls apart, 2 polypeptides are made. The peptide bond is broken. So because this guy was unstable, the most unstable part of it turns out to be the peptide bond, and that falls apart. Yes, Cassandra?
Is the Oxyanion Hole what stops it from just reforming again? When that bond just got broken, just reform?
Very good question. So her question is, does the Oxyanion Hole prevent this thing from reforming? And the answer is basically no. Okay? Because once the bond has been broken, there's nothing to hold it in place. And this literally just goes flying out of there. That's why this is the fast phase. So if you have a chain and you have a variety of links and you break one of the links, if there's something pulling on it, they're not going to stay together. And that's basically what happens here. It just goes out. You've broken a link in the chain, and this guy is now going away. Okay? All right. Good question then. We have an Oxyanion Hole down here. Same oxyanion hole, and it's there for the same purpose. We don't want this guy to react with the enzyme. In this case, the bond that we're breaking is this one. The oxyanion hole stabilizes it and lets it fall apart. And when it falls apart, this bond gets broken. So the role of the oxyanion hole is to stabilize an unstable molecule and keep it from making a covalent bond with the enzyme. The result of that stabilization is the molecule falls apart. So I said there were two pockets. One of the pockets is the oxyanion hole. The other pocket turns out to be the binding site for the proper amino acid. The binding site. Let's think of that phenylalanine this thing cuts next to. It turns out there's a little pocket that phenylalanine sticks itself into when it comes into the enzyme. That's how the enzyme knows it's got the right thing. The side chain of the phenylalanine amino acid is sticking itself into this other little pocket, and that other little pocket is called the S1 pocket. The S1 pocket has some very cool things I'll talk about next time, but that's what's going on here. So the S1 pocket is the binding site for the amino acid side chain. It's right there, a little pocket off of the active site. Okay? Now, would you like me to go through the mechanism one more time? Okay, yes.
I have a question about the oxyanion hole.
Yes.
Is it actually a space, or is it just a bunch of negatively charged R groups that are sort of repelling and negative?
Well, it's some side chains that are of amino acids of the protein that are not reactive. That's a critical component. And I can't tell you if they're plus, minus, or whatever. They're not reactive, and they keep the reaction from happening with the enzyme. So they're counter to that reaction with the enzyme. Yes, Laurie.
So is the S1 pocket essentially an identifier that doesn't get full, the reaction doesn't happen, and the enzyme knows that it has the wrong amino acid?
That's correct. So the binding in the S. Her question was: is the S1 pocket sort of an identifier? If things don't bind there properly, does the enzyme then not go through that conformational change? And the answer is that's correct. So you could say, well, what if you bind a tyrosine instead of a phenylalanine? Okay. Well, it turns out that the electronic environment of that S1 pocket is such that a tyrosine won't fit in there. Well, I'll explain that probably next time, but you'll see more about how you can change the S1 pocket and actually change the specificity of the enzyme. That's kind of cool. Okay? I see another hand? Okay. All right, let me go through the mechanism one more time. So, I've got chymotrypsin. Chymotrypsin is sitting here. It doesn't have anything bound properly. If it binds to a polypeptide chain and the wrong thing goes into the S1 pocket, the enzyme doesn't do anything. It just lets go. In fact, it won't even fit in the S1 pocket unless it has the right shape and charge. Okay? If the proper thing, however, is there and it fits in the S1 pocket, it causes the enzyme to undergo a slight conformational change. That slight conformational change results in changing the geometry of the catalytic triad. The three amino acids are moved closer to each other. The first effect of that is that the negative charge on the carboxyl side chain of aspartic acid comes closer to this ring and essentially pushes electrons to the right as you see it, making that nitrogen electron-rich, slightly negatively charged. This slight negative charge on this nitrogen attracts the hydrogen of the side chain of serine, removing it and creating an alkoxide ion. The alkoxide ion is very reactive, and it attacks the peptide bond of the polypeptide that's sitting there. Okay? That creates an unstable intermediate that you see here that is stabilized by the oxyanion hole. And that stabilization is important to keep this reactive intermediate from reacting with the enzyme itself, which would kill it. So instead of reacting with the enzyme itself, the polypeptide falls apart, and it falls apart at the peptide bond. There's the link in the chain that's broken. This R2 component essentially falls out and grabs the hydrogen that was there as it goes, taking the hydrogen with it. You're left behind with the other portion of the polypeptide chain covalently linked to that oxygen on the side chain of serine. So what you see up to this point is we've gone through the fast phase of the catalysis. The rest of this is the slow phase of the catalysis, and it takes longer, time-wise, for this to occur. This bond has to be broken to release the other polypeptide chain and allow the enzyme to go back and catalyze breakdown of another one. To break this bond of the other portion of the polypeptide chain with the enzyme requires water. Water comes into the active site, and something similar to what we saw before happens. The negative charge in this ring, which is now negative again because notice that hydrogen is gone, attracts the hydrogen off of this amine group and creates a reactive hydroxyl group. This reactive hydroxyl group attacks this carbon, causing an unstable intermediate. The unstable intermediate is stabilized by the Oxyanion Hole. It falls apart instead of reacting with the enzyme, and that causes the release of the second polypeptide. At that point, we're back to the state that we started. Okay. I see fewer frowns this year than previous years. Usually the whole class is totally frowning, so I think that's good. Questions about that? Yes?
I understand the mechanism. I'm missing the big picture. Like, it's just cleaving the peptide.
Okay. So his question is, he doesn't see the big picture. What's the big picture here? The big picture is, this is how it cleaves a peptide bond and releases pieces. And we're seeing step by step at the molecular level what's happening with that.
It's going on in all the cells.
It's going on whenever we have this enzyme active. Okay? Now, one of the reasons I show you this mechanism is because the mechanism that we see here is a common mechanism of all serine proteases. We see there's other enzymes. Trypsin, for example, is a serine protease. And when we look in trypsin, we see the same catalytic triad. We see the same arrangements of things in the active site, aspartic acid, histidine, and serine. And we see exactly the same mechanism. The only difference is trypsin has a different S1 pocket. And so it binds different amino acids, but it does the same thing. It's breaking those bonds, cleaving that peptide bond adjacent to different amino acids. And I'm going to go through some of those as I have time. Other questions about that? Yes?
The S1 pocket isn't shown. Should I just imagine the R1s
Yep.
being attached to it?
That's correct. The S1 pocket is not shown for simplicity's sake. Okay, but you can imagine that that the side chain of phenylalanine is sticking somewhere out there in that pocket, as it were. Everybody got it? I'm not in a hurry. Yeah, Laurie?
So the hydroxyl ion attacks the carbon outlet bond and ends up the carbon floating away.
Wait, say that again.
In between step 6 and 7.
Between 6 and 7,
That reactive hydroxyl
attacks this bond right here.
attacks that bond, but then ends up going away with that piece.
It goes away with that piece. That's correct. That's what's shown right there. Okay, and so we see that the hydrogen, okay, that came in with that water is what gets left behind here. That's not a big point, but you can see that the hydrogen that we start with is different than the hydrogen we end up with. Again, not a major point at all, but for anybody who's tracking hydrogens, that's what's going on. Yes, ma'am.
Is it possible to do this reaction in some solution other than water and get stuck halfway?
Is it there's an interesting question. The question is it possible to do this reaction in a solution other than water and get it stuck halfway? I'll ask you guys to predict that one. What do you think? No way over here. How many no ways? There's a no way. Real. How many say yes? Okay. All we can do is argue, because we don't have this set up here. But I would argue with the no ways. And the reason is that you're unlikely to have the properly folded enzyme if you put it in a non-aqueous solution. Because what you'll probably do is you'll unfold it. I can't tell you that definitively, but I suspect that's what would happen. Enzymes aren't very tolerant of non-aqueous environments. Okay, well, let's move forward. And we're not too far from done for the day. Okay, so all these steps are just close-ups of each individual thing there, so you don't have to worry too much about that. There's the Oxyanion Hole. You can see the configuration of it. There's a glycine there, okay, and other things that are stabilizing that. The bottom line is it's keeping the enzyme from reacting with the substrate. And I'm not going to worry about that for the moment. And I'm not going to worry about that for the moment. So don't worry about the geometry as you were asking about. Is this on the R1 side or R2 side? That's not the big picture. The big picture is the mechanism itself. And so don't even worry about this figure. Now, I said that there are other serine proteases, and there are many serine proteases, and the one that we've talked about already is trypsin. Trypsin, if I look at the three-dimensional configuration of trypsin, and I forget which one's which, but we see one's in red, one's in blue, we see some great similarities between the two. And one of the places where the two enzymes differ in their structure is in the configuration of the S1 pocket. Chymotrypsin tended to prefer to have the nonpolar amino acids. Trypsin, if you recall from memorizing that from earlier, tends to prefer to cut next to lysines and arginines. What's the common feature of those two side chains? They're both positively charged at physiological pH, right? Okay. Let's look at the S1 pockets of some different proteases. Here's chymotrypsin. It's a fairly nonpolar S1 pocket. You don't see charged groups in here. This allows non-charged, nonpolar side chains to fit in there and be comfortable. Here's trypsin. Look at the bottom of the S1 pocket. There's a negative charge attracting anything that's positively charged and repelling anything that's negatively charged. And the nonpolar stuff doesn't have a happy environment. It doesn't want to get in there. This is why trypsin cleaves at lysines and arginines. Elastase is another serine protease, and it tends to cut adjacent to amino acids that have a small or short side chain, like alanine. And that's because it's blocked at the bottom. It has a very small pocket. It won't accommodate something bigger. Structure is necessary for function. Keep coming back to that. Okay? Structure of an enzyme is critical for its function. If I disrupt the structure of the enzyme, I'm going to disrupt the function of the enzyme. If I change something in the S1 pocket, I'm going to change something about the specificity of a serine protease, because it's going to determine what binds in there and how well it does. Okay? All right. And one last thing, and then we'll finish. This is simply to show you a completely different serine protease from what I talked about before. This is a protease called subtlysin. Subtlysin, look at what we've got there. We've got a serine, we've got a histidine, we've got a aspartic acid. And just because they've drawn it left to right doesn't mean it's any different. It's not. Left and right to left are equivalent. It's got an Oxyanion Hole, just like the other one did. So the bottom line is that this structure that we see inside of serine protease is critical for its functions, and we see it conserved evolutionarily across many different proteins. This is a very functional sort of structure. Yes, sir?
So I can only assume that the digestive system has quite a few of these proteases.
Correct.
What stops them from destroying each other?
Okay, so his question is, he can only assume that we have plenty of these proteases in the digestive system. What stops them from destroying one another? Nothing. Okay? Now, I'll talk, that's a big simplification. They do destroy each other. But we'll see that what matters is not they're destroying each other, but it's very important that they not destroy tissue that makes them. Cells have proteins in their cell membranes. And if the cell makes a protease and the protease starts digesting the cell, that could be a problem, and it's a big problem in some cases. And that takes some control, and I'll talk about that later, but it's a very good question. Okay, let's do that. Remember when you come in to position yourselves appropriately, get here in plenty of time to get the exam started, and I'll see you guys on Monday. I'll see you actually at a review session tomorrow. Yes, ma'am.
I've heard a lot of people that have taken this
Yeah.
a good place for me to start?
That's always why.
I'm starting at point 1 right
That's
now.
always where I say to start. So start the highlights and then just branch out from there. But the highlights are always what I think were the things I talked about that were the most important.
Okay.
So absolutely.
Good starting point. Okay.
Absolutely.
I will
Okay.
start tonight.
Study hard, Cassandra. Yes, ma'am.
I just want to make sure. So all serine proteases have in common an S1 pocket
Yep.
that helps make sure that the right substrate binds and the right thing is in the right place at the active site.
That's the specificity part. Yep.
Then they all have interactive site, the catalytic triad of serine